Good morning, everyone. Maybe we should get started with the scientific program. So good morning, I'm Phil Beales. Lovely to see you all again, and several new faces. Actually, could I just ask you to put your hand up if you've been to this conference before? Oh, that's a majority of you. Okay, and if this is your first conference, hands up, please. Oh, wow, that's a huge number. Well, welcome. So I think what we should do then is introduce ourselves. So if you just spend a minute, if you turn to the person on your left and then the person on your right, just say who you are and say good morning and welcome. Okay, steady on. Let's not get too familiar. Okay. All right. You can carry on afterwards. That's the idea. All right. Okay, just want to quickly run through the program for this morning, so that you're all aware of what's coming, and after I've given you a little bit of an update on research that's going on at the moment, we're going to actually hear from a staunch supporter of ours, Helen May Samira, who's come from Germany today, and she's going to tell us about the research program that she's been undertaking back in Germany. We're then going to hear from Dr. Murray Stewart, who's telling us about a new study, and I'll leave that to him to talk about. I will introduce it though. And then we're going to hear about how the clinics have been developing and the advantages and developments in that as an invaluable resource from Dr. Shella Mohammed, who's a consultant in clinical genetics at Guy's Hospital. And then after the break, we're going to hear from Kerry Leeson-Beaver from the Alström Syndrome UK, who's going to tell us about breaking down barriers. Ellie Southwood from RNIB is going to be here to celebrate 150 years of RNIB, which is amazing, about 6 times as many as the BBS UK. We're going to hear from Sally Flatto-Taylor about the importance of whole family support. We're going to hear about the first personal perspective from Maria Dowswell. Then we're going to hear from Amanda Avery from Slimming World. And finally, the second personal perspective from Tom Oates, and then we'll break for lunch. So is everyone happy about that? Yep. Okay. So, right. So I'm not going to take very much of your time because we're under starters orders. But essentially, as you all know, we've been running the clinics now for 10 years. So half the time that a third of the time that this organization has been in existence. And Shella's going to tell you more about the structure of this, but I put up this what I call the Wheel of Fortune, which shows all of the partner organizations that are involved in delivering the clinic. So as you know, we have 2 centers, 2 hospitals in Birmingham Children's and University Hospital Birmingham, and we have 2 in London as well, Great Ormond Street Hospital for Children and Guy's and St Thomas's. But they're supported, I need to change this slide from LMBBS to BBS UK. Tonia's just spotted the deliberate error there, but ably supported obviously by this organization. But also, when we first set this up, we also developed the first genetic testing center in the world for this particular condition. And that has been extremely beneficial going forward, because we've now completed, this is a bit out of date actually, a few months out of date, but we've completed almost 800 genetic tests, okay? And there are 21 genes, or there were 21 genes, I'm going to tell you about a couple more actually, that we know about. And you only have to have spelling errors, 2 spelling errors in any one of those genes to manifest, to have the problems associated with BBS. So out of those tests that we've done, about 60% of them are actually patients and the rest are the relatives, okay? So we support relatives as well who need to have carrier testing, want to have carrier testing within families. But I'm really amazed now to say that we're beyond 80% in terms of the number of people who were able to provide a genetic test for where there is a positive confirmation of the syndrome. Because that's really, really important for a number of reasons, retrospectively for us to go back and understand whether there are differences according to the different gene mutation that you might carry, but also in terms of thinking going forward in terms of the kind of treatments that some of which, as you'll hear potentially later, may be related to the type of gene that happens to be involved. We've done over 20 what we call predictive testings. That's tests on prenatal, in the womb, if you like, or on a newborn baby to determine whether or not they're going to be affected in advance. But that still leaves a significant number that don't actually have a genetic mutation. So there are a few that still need to be discovered. But I really do have to thank Beth, whose birthday it is very soon, 40th birthday. Really, I'm really serious here, because Beth has been with us for over 20 years. She did her PhD in my lab donkey's years ago, but she really is the person who has driven the genetic testing behind this service. Okay, so we have to thank a lot to her for developing the tests and making them happen. So thanks very much, Beth. So in the literature, in the last couple of years, there are now a couple more genes. So we're now up to 23, I believe. And another one just a few months ago called SCAPA. Don't know what it does. But essentially, it's another one that we would add to the list. But what I'm saying here is, the reason I'm telling you this is because we're going to have to change our test accordingly. But we're also at the point where we're scraping the bottom of the barrel, really, because both of these particular situations, we only see like one family which has those particular genetic changes. So they're not going to be accounting for a lot. So I don't think we really have to worry too much when it comes to the design of our test going forward. So let's just talk a little bit about some of the things that have been happening on the research front, worldwide that is, that are relevant to you. So I've put together, I haven't done this for a while now, but I've put together a little graph here that I'm showing of all of the papers that have been published since 1949 on Bardet-Biedl syndrome. So in 1949, there was one paper. Then it waxed and waned. We didn't really see much until about 1995. Then it started to increase when people got a bit interested. Then it shot up around about 2000 and early noughties, which coincided with really when we found the first gene for Bardet-Biedl syndrome. And then you can see there's been a real sharp increase after 2005. And that really correlates with when we first started to understand what the function was of the genes. And that function, as many of you will know from previous visits, is around how the cilia work in the body. And we can discuss that at any of the workshops this afternoon, because I'm not going to mention that again right now. So we've been doing pretty well in terms of papers. I mean, in probably 2013 is the high point, where there were about almost 70 papers published in that year. And this year we're up to 27, and we're only a third of the way through. So we should be on target to get up to about 60. So what I'm saying is that there's still a strong interest in BBS, and that's up to us to maintain that awareness and interest in the medical community and the scientific community going forward. So what about some of the papers that were published this year that might be of interest to you? Well, one of them from Bob Hawes, who's a leading proponent of BBS from the Marshfield Clinic in the US. He's published a paper about skin problems associated with BBS that some of you might recognize. So his paper is entitled Cutaneous Findings in Bardet-Biedl Syndrome, published in the International Journal of Dermatology. And he essentially determined that there were a couple of conditions, skin conditions, which are common conditions in the general population, but they're much more common amongst folks who have BBS. And one of them is called keratosis pilaris, and the other one is seborrheic dermatitis. Now, seborrheic dermatitis is very common. It leads to scaly and itchy skin, particularly in your scalp, but it can happen in other places. Sometimes looks like psoriasis. And it's quite difficult to treat. But it's a bit like a severe form of dandruff, if you like. But the second one, which 80% of you seem to have, is something called keratosis pilaris, which is actually a sort of a mild rash, but it's an itchy rash. Sometimes looks a bit like eczema, but it's not. And what it's due to is a buildup of keratin. So keratin is a protein that sits on the surface of our skin to help us be waterproof. And what happens is it seems that with having BBS, you produce too much keratin, and this clogs the pores, and then you get a rash. And so sometimes it's described as chicken skin, or sort of like multiple pimples. And again, this is treatable, but again, it can be troublesome. But this is something I thought I would, alert the community to, because you might want to discuss that at some point and think about some of the treatments for those things. Okay, so I'm going to just really touch on some of the research that we presented in years gone by. So, for example, Victor Hernandez, who's here, where's Victor? There we go, has been leading the charge in terms of gene therapy. And I know a lot of you are sort of tired of hearing us go on about gene therapy, and I'm not going to go into the details at all, because again, Victor's going to be around later to talk about this. But just to say that we've actually got to the point now where we've completed successfully, I would say, the first stage of the gene therapy studies. And just to remind you, there are 2 gene therapies that we have been developing, and we've been developing this in what we call the preclinical phase. So we use mice that have BBS. They have all the similar problems with visual impairment and obviously weight gain. And what Victor has developed is 2 gene therapies, one for the eye, which is a subretinal injection of BBS1. So you're replacing, or adding to, should I say, the faulty BBS1 in the back of the eye. And I'm happy to say that this study has now shown that this actually rescues, or rather prevents, blindness in those mice that are treated. So we want to take this forward into the next stage in clinical trials. The second one, again led by Victor and his team, has been in partnership with a funding agency called Apollo Therapeutics, and this is developing a gene therapy, a more systemic gene therapy. So what do I mean by that? I mean where we've tested, again in mice, the delivery of a gene therapy through the vein and directly into the brain, okay? And the reason for that is because, as you all know, BBS affects many parts of the body, many different organs, probably all of the organs, to be honest. And therefore, is it possible that we could develop a gene therapy that can really reach all the parts of the body that we need to reach? So this is the purpose of this particular study. And very briefly, the outcome of this is that this form of BBS1 gene therapy, given either intravenously into the mouse, but particularly into the brain, into the ventricles of the brain, actually prevents obesity. So this is essentially the first gene therapy out there that at this early stage can actually prevent a significant amount of obesity. And that, I think that is going to have potentially a huge impact. So we're at the point now where we think we're fairly close to getting some funding to take this to clinical trials. So hopefully I can come back to you next year and tell you whether we have got that funding and that what the structure of that particular study might look like and the course of time over which it will be to actually deliver that particular study. So, moving on. So again, some studies you've heard about in the last coming years from Sonia Christou and also Naila Haq, my PhD student, have been working, and again with Elizabeth as well, Elizabeth Forsyth, working on various aspects of the brain. But what Naila and Sonia have been able to do is to be able to start to dissect what is it in the brain that's starting to lead to the need to eat more or to put weight on. And so essentially what you need to understand is that the brain doesn't operate in isolation, it operates as a complete unit with our gut. So I'm showing you here a diagram for those of you who can't see it, which at first sight looks complex, but it's actually quite basic. You've got the brain on the left-hand side, you've got the gut, you know, the intestines, small intestines at the bottom here, and on the right-hand side, you've got the fat stores in the body. And those are the three things that speak to each other. So the gut is at the center of this because when we eat, we have to then tell ourselves when we're full. And that message has to go to two areas. It's got to go back to the brain to tell us to stop eating anymore. And it also has to signal to our fat stores where to put that energy, that excess energy, if we particularly want to have the extra packet of chips or what have you. So what happens is that the fat stores send out a signal, which was discovered about 20 years ago, called leptin, which signals back to a part of the brain, the appetite center in the brain, called the hypothalamus. And what we're now beginning to understand is that the BBS proteins are really, really important for the part in the brain where that signal is received. And we think that it's not working quite as well, it's blunted. So therefore there is a resetting, if you like, of the threshold that says, right, I'm not quite full enough yet. Okay, so then the signals are also coming from the stomach. And they're coming in the form of a hormone called ghrelin. So I don't want to complicate the picture, but there are two real important proteins here, ghrelin and leptin. So ghrelin is coming from the stomach. As soon as the food hits the stomach and starts stretching your stomach, ghrelin will actually go down. Okay, so it's produced by the stomach when you're hungry, before you eat. But leptin is low at that point. After we eat, or during eating, ghrelin goes down in the stomach, and the leptin goes up. And so this is all about the balance. And so what's been happening in our lab at the moment, between the work that Sonia and Naila have been doing, they have worked out the process, the feedback mechanism that's coming from the stomach up to the brain. And again, so it's not just about what's going on in the brain, it's also about what's going on in the stomach, or at least the signals that are going from the stomach to the brain. It's early work, but again, it's an area that I think we need to be developing, and we've got grants in, we're waiting to see whether we're successful on that, to see whether we can develop more work around that particular area. So then really my last slide is to tell you about, or really to introduce a new study by Rhythm Therapeutics, and you'll hear a little bit more from Murray in a moment. Tell us about the new setmelanotide study, which is based particularly on those feedback mechanisms that I told you about, particularly in the brain. And I'll leave Murray to tell you about that shortly. Okay, that's my time up. We've decided that we don't really want to take any questions this morning, but I think we might take one question, should we say. But because everyone who's talking this morning is going to be doing a workshop, so if you can save your questions, your burning questions for those, that would be great. I think we're okay on time, so I'm happy to take one question, I'm told. And has anyone got a question?
The treatment you've got for BBS1 sort of getting quite far along. How hard is it to move a BBS1 treatment, on BBS10 or 16?
Yeah, it's very difficult. So the question was, if this is for BBS1, what about the other ones like BBS10? And we can talk until the cows come home about, we did start to develop a BBS10 study as well, but for various logistic reasons, it didn't work. And we can talk again of why that failed, but it doesn't mean we're not going to do it. But what we're doing is we're focusing on BBS1 because the challenge now isn't actually doing this, isn't developing the study, it's actually getting it funded. Okay, we're talking about a need to go to the next stage, the first clinical trials of about £3 to £4 million. And that's just for BBS1. But you're quite right, we do want to do BBS10, because if you do BBS1 and BBS10, it covers almost 65% of the BBS population. So in two fell swoops, should I say, we should be able to significantly change that. But there will be other treatments coming along, and that's why Murray's been invited along today. Okay, so I think we'll move on to our next speaker, if I may, who's Helen May Samira, who's come from the University of Mainz.